Okay, so now let's discuss about some methods of gene therapy. So we have already discussed about the classical gene therapy and non-classical gene therapy. So in this video we will going to discuss about a approach by which you can transfer your gene inside a cell which either contain some defect due to a particular gene. Okay, so that kind of gene therapy is ex vivo gene therapy that I want to talk with you. That means ex vivo gene therapy. What is the meaning of that ex vivo gene therapy? So the term ex vivo means here we are doing something outside the cell, right? Doing something outside the body, right? So here that simply signifies that we are going to transfer the gene inside the cell outside the body. Okay, that is the meaning of the ex vivo gene therapy. So what we are doing here, so here suppose, and another important thing, that ex vivo gene therapy is only and one and only applicable for those cell types that can be easily isolated. Okay, that means for a delicate internal organ which is present in too much deeper part of the body, we cannot going to use ex vivo gene therapy for that purpose, because in those cases we will going to use in vivo gene therapy that I will going to discuss in the next video, where I will discuss with you about how the gene can be transferred directly into the cell inside the body. Okay, so now in ex vivo gene therapy, that means we can only perform ex vivo gene therapy for those kind of cells that can be easily isolated from the body. So the most common two things, that the two kind of cells that can be isolated easily from the body are, one is our skin, okay, and another one is the hematopoietic cell. Okay, so hematopoietic cell means we are here talking about immune cells, mainly neutrophils, stuffs. Okay, so now we can easily isolate them from our blood serum, and we can isolate skin from directly, right? So suppose the defect is here in skin. Okay, so first of all you have to collect the patient cell. Okay, now you will grow them in an appropriate culture medium. Okay, so now you will going to use a vector system for transferring your gene of interest inside those cells present in the petri plate. Okay, so according to this picture, the therapeutic gene is inserted in an inactivated virus, and then virus is used as a vector for transferring. Okay, so now after transferring, the therapeutic gene containing cell can be easily selected. Then those kind of cell will be further multiplied, and then we will going to transform them back into the individual's body. So here we are using patient's own cell. Remember it very carefully. Here we are using patient's own cell. Why? Because if we not use any kind of autologous cell, then as a result what will happen, our immune system or the patient's immune system will show rejection towards those newly introduced cell. Okay, and as a result our entire curing process will be failed. Not only that, it can also invite various kind of complication inside the patient's body also. That is why choosing of autologous cell is important for ex vivo gene therapy. That is why it is often preferred to use the patient's own cell for that ex vivo gene therapy. Okay, and remember another important thing, that ex vivo gene therapy is only used for those kind of cells that can be easily isolated. For example, tissue containing skin cell or hematopoietic cell. Okay, now after isolation of those cells, we will grow them in a culture medium. Okay, so now we will cause the transformation of the therapeutic gene with the help of a inactivated virus. or we can also use another kind of system also. Okay, so after the transformation we will select only those cells that have acquired the therapeutic gene. Okay, so now after selection we will selectively multiply them and then we will transfer them back inside the human. Okay, and inside the human, due to the expression of the therapeutic gene via various kind of process, the disease cell can be cured. Okay, so that is all about ex vivo gene therapy. So here I have mentioned all the 4 important points that I have explained diagrammatically. That is, in this method of gene therapy, gene are transferred to the cell grown in culture, and then transformed cells are selected. We have to select them with appropriate antibiotic or anything that we have incorporated for selection. Then we have to multiply only those selected cells sorry, and then introduce into the patients. Okay, the use of autologous cell is very important to avoid immune cell rejection, that is I have told you. Okay, now the cells are sourced initially from the patient to be treated and then grown in culture and then being reintroduced into the same individual. Okay, now this approach can be applied mainly for hematopoietic cell and skin cell, because they can be removed from the body and they can be then genetically corrected and then outside the body and then reintroduced into the patient body, where they become engrafted and survive for a long time. So that is the idea, simple idea, that we have to transfer our therapeutic gene inside the cell when the cells are grown in a culture, and we have to use only autologous cell for doing the ex vivo gene therapy to avoid immune cell rejection, right, or immune system rejection of the introduced cell. Okay, clear? And here we are using any kind of vector technique for transfer of gene of interest into the cell. Okay, so that is the video for ex vivo gene therapy. So in the next video we will going to discuss about in vivo gene therapy.